Hey everyone, welcome back. All right, so we're going to talk a little bit more about neurons and how they work, and really now we're going to focus in on some neurotransmitters um, and basically give you just a very, I don't know, I guess broad overview of some of their functions. Right? These can get super specific. There are entire disciplines, journals dedicated to each one of these. We're not going to get anywhere near that depth. Again, just very superficial level, just to give you an idea of some of their functions. Okay, so with that, let me just kind of give you a little bit of a, I don't know, tidbit kind of fun fact. What you're looking at, these are actual images taken using microscopes. They're actually using fluorescent microscopes to take images of neurons in the brain. So what they've been able to do is actually use principles of the immune system, so antibodies, and they're able to tag very specific receptors on the surface, or proteins I should say, on the surface of the cell membrane of different neurons. So depending on the cell that you are, you'll have slightly different proteins on the cell membrane, and they'll make antibodies. They'll color it a different color, so specific cells will light up a specific color. So you can see basically all the ones that are in green are one type, the ones in blue are one type. Red is a totally different protein, different fibers. So axons and dendrites will have different proteins expressed in them. So you can actually tell difference now between axons and dendrites, something that we couldn't do in our lab, right? They all look the same because our stain wasn't specific enough to label them differentially. You have the same kind of thing here. You can really see the axons and dendrites. One is in red, the other is in green. And then here, what you're seeing is like this. This is actually an image, again, taken from a microscope. And what you're seeing here is that depending on the age of the cell, it expresses a slightly different. They express different proteins. And what they've been able to do is color code all these different proteins. So now you know that all the greens were born at the same time, all the purples were born at the same time, all the yellows were born at the same time, and kind of where they've then ended up distributing themselves. Okay, so just, I just thought that was kind of a neat little fun fact about where we've gotten with some imaging and what neurons kind of look like. All right, so let me hide me, or maybe I can just actually just move myself here. All right, so we had talked about neurotransmitters a little bit, right? So again, we had talked about acetylcholine when we talked about muscle contractions, and in the previous video, we talked about how the neurotransmitters are released at the synaptic end bulb to get across that synaptic cleft, so that the signal can get transmitted to the next neuron. All right. Well, what do we do with that neurotransmitter that's just kind of floating around in that synaptic cleft? Well, some of that simply just diffuses away from the synaptic cleft. So you dumped it in a space, some of it just kind of, wanders off and eventually gets cleaned up by some of the microglia and other phagocytes. There are also other enzymes that will help degrade the different neurotransmitters, right? So you've probably maybe you've heard of some of them involving serotonin, for example. We've actually seen an enzyme already, when we talked about muscles. You remember what that enzyme was? So we talked about acetylcholine, right? That was our neurotransmitter. So the enzyme that broke down acetylcholine in the synaptic cleft was acetylcholine esterase. Okay, so it helped, again, kind of clear it from that synaptic cleft, allowed for more specific signals sent to muscles. And then finally, some of that neurotransmitter is simply taken up by cells and recycled. So kind of like this little recycle bin truck right here. So another cell, usually an astrocyte, sometimes the cell itself, the presynaptic neuron, will simply take up any of the excess neurotransmitter, gives it back to that presynaptic neuron. That's about it. So you recycle them. And you probably have heard of SSRIs. You may have heard of those. So those stand for selective serotonin reuptake inhibitors. So that'd be an example of where we inhibit the reuptake of serotonin. And by inhibiting the reuptake, more serotonin stays in that synaptic cleft. So instead of having to give someone like a pill that contains it, we simply leave it in that synaptic cleft longer so it can do whatever it is going to do. Let's see. All right. So we're going to talk about two different concepts. One is excitatory postsynaptic potentials and then inhibitory postsynaptic potentials. So in an excitatory postsynaptic potential, basically, let's just kind of read this. So a neurotransmitter that depolarizes the postsynaptic membrane is excitatory. Okay? So that's all that means. So here's our presynaptic neuron. It releases the neurotransmitter to that synaptic cleft. It impacts the postsynaptic neuron, and it'll excite it. It'll depolarize it. So that means that if another signal comes in, it's more likely for this thing to fire. Okay? Does that hopefully make sense? So again, that postsynaptic potential is excitatory. Usually where we see these connections, because remember, neurons can make connections all over the place. But basically, if you have the axon of one neuron contacting the dendrites of that postsynaptic neuron, that connection implies that it's going to be an excitatory signal. Okay, just the connection alone. Not always, but chances are that's what it's going to be. Okay, I don't know, it's sort of like, it doesn't matter what car we get into, we assume that, the clutch is, way to the left, gas is way to the right. So we don't have, so basically the same kind of idea. We can always assume that depending where that connection is, it's going to be excitatory. Okay. All right. So and then on the other hand, so kind of going along with this car analogy, if we had something that caused the, sort of like the accelerator, we're going to have a gas pedal. So we also have inhibitory postsynaptic potentials, okay? So in this case, a neurotransmitter will cause hyperpolarization. So meaning instead of getting closer to that negative 55 threshold, we go in the opposite direction. Maybe we go from negative 70 down to negative 80 or negative 90, right? So it'll cause hyperpolarization of that postsynaptic membrane. And because it's going away from that threshold, it's just that much harder to get that postsynaptic neuron to fire. So it's kind of like the brakes on a car. Okay. So again, it's just what I said here. So during hyperpolarization, the generation of an action potential is more difficult because that membrane is just that much further from the threshold. And then usually where we see that kind of happening is when the presynaptic neuron is making contact with the cell body of the postsynaptic neuron. Again, that's sort of where the brakes are. Okay. And then ever so often, so this little note down at the bottom, ever so often you'll see the synapses on axons, so axon to axon. That doesn't inhibit, that doesn't accelerate or excite. Basically what that does is it simply modulates the amount of neurotransmitter being released. So you've already gotten the excitatory signal from somewhere else. This sort of tweaks and says, okay, and let's make that a little bit more, or let's make that a little bit less. Okay, that's all that is. So if it's making axon to dendrite, excitatory. Axon to cell body, inhibitory. Axon to axon, it's going to modulate the signal. All right, I'm going to move myself here. So basically, we're going to take this idea of excitatory postsynaptic potentials, right here, it's abbreviated EPSP, and inhibitory postsynaptic potentials, IPSP. I'm going to put that together, okay? So basically, like you can see here, so neurons are going to, I know when we looked at it in lab, it didn't look like a lot of connections, but for some reason it's a really weird thing, and they don't really understand why still to this day. When you stain something, you don't actually see all the neurons. We're getting better. So you saw like the images that I showed you at the beginning of this presentation. We're getting better, so we actually are getting to see the majority, if not all of the, I don't say all, but the majority of the neurons now in the brain region. But in the past, we wouldn't, like about 1/10 of them would stain. Anyway, that's what we saw in lab. However, a typical neuron in the central nervous system will contact, or get contact from, I should say, anywhere from 1,000 to 10,000 other synapses. I mean, that's a lot of connections, right? I mean, I don't know, that'd be equivalent of a ton of friends on social media, right? On whatever social media site that you're using, right? So, and as you can imagine, just like you getting input on that social media website from 1,000 to 10,000 individuals, that might influence how you react to something. We go with this analogy. So let's just take a look here. So here we have our postsynaptic cell. It's in this sort of beigey color, right? And then we have presynaptic neurons. So we see these like they're contacting the dendrite, dendrite. And then here we're a little bit more on the cell body. And you'll see this one is going to be inhibitory. So what ends up happening, right, is these are going to be excitatory. You see a little, it's going closer to threshold. So we're depolarizing the cell membrane, depolarizing, hyperpolarizing, right? Now, we have two ways that this can then trigger an action potential in this postsynaptic cell, right? So one way is spatial summation, and that's really what you're seeing here. So, this axon, is telling this cell to excite, inhibit. Well, that's 3 to 1. It's probably going to excite, right? It's probably going to fire. So that's spatial summation. So in space, you have many things telling it to do one thing, and then very fewer of the other telling it to do the other, right? Sort of like, I don't know, 100 of your friends tell you to do something, and then 2 of them tell you not to. You're probably going to do it, right? Because you got the input is overwhelmingly toward doing it, right? So similar to here. So as long as you get the excitatory postsynaptic potential, it'll take you closer to threshold, closer to threshold, closer to threshold. One inhibitory, another threshold, you're going to hit it. Hopefully that makes sense. Temporal summation, on the other hand, is a little bit different. It isn't so much, well, it isn't at all, how many are coming in. So it isn't like 100 to 1 kind of thing. Instead, what it is basically one neuron rapidly firing, going fire, 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 fire. So excitatory. So with that many incoming small excitatory postsynaptic potentials, you're going to eventually hit threshold. Okay, so basically the difference is spatial summation. You have many coming in, sending their signal, and either this neuron, in this case, in the image here, it will fire, right? Because you have more excitatory than you do inhibitory. In the case of temporal, it would be a 1-to-1 kind of thing. It's just that this one's going fire, 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 and then the postsynaptic goes, okay, I'll fire. I kind of think of it as, I guess my analogy again, so spatial summation reminds me of that social media, again, that like 100 friends telling you to do something, 1 or 2 telling you not to, you're going to do it. Temporal summation is more like your significant other nagging you 100 times to take out the garbage. It's just one person doing the nagging, but they do it enough, you're going to eventually take out the garbage. Okay, so that's sort of my analogy. All right. We have this guy, Donald Hebb. It's a really important key figure in neuroscience. He came up with this tenet called Hebbian plasticity. And this is, in this second bullet point is actually what he said. But in this third bullet point is what it means. And this is how everyone in neuroscience remembers it. Basically, it says neurons that fire together, wire together, all right? And what that means is that if we have this neuron here, and it is constantly sending a signal to this neuron here, saying, hey, do it, fire, 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 and this one's reacting to that, what ends up happening is eventually they'll build a much stronger connection between the two. Maybe you have several synaptic end bulbs from, this one making contact with this one, right? So the connection becomes stronger. Versus if this neuron, let's say we get another set of neurons, I don't know, pinkies, different set of neurons. So let's say we have this one and this one, they have a connection. This one's ever so often like fire, fire. It's not a very strong signaling, right? They don't really fire together. That connection will eventually go away, okay, or it'll be weak, okay? So again, the more that one neuron sends that signal to another, the stronger like their physical connection becomes to one another. It makes sense. It's sort of, again, let's use social media or just friendships. The more time you spend with someone, the closer friends you usually are, and that connection's gonna be really strong, right? And that's similar to how our brains are. The more we study, the more we work at something, the more we practice, and that's gonna be really key. So the more you practice, the more times those neurons are sending signals to one another, the more times they're connecting, that connection becomes really strong, right? So think about this, the stuff that you've practiced like over and over and over again, even if you don't do it for a while, you come back and that connection's still there. You still remember how to do it, right? Because you built up a nice strong connection. Versus if you only look at it once or you read it once or you do it once, You might remember it for a short time, but then that connection goes away because you never really solidified it. Hopefully that makes sense. So again, neurons that fire together, send signals to one another, are going to wire together. And again, the more they fire together, the more they influence one another, the stronger that physical connection is going to be. Super key tenet in all of neuroscience. All right, so I'm going to hide me here so you can see this better. We're going to talk mostly about neurotransmitters. I just wanted to bring up the concept of a neuropeptide really briefly. You may encounter these in your studies. And just so you know what these are, essentially they're going to be the same thing. So neuropeptide is basically the same thing as a neurotransmitter. However, it's a smaller protein molecule. That's all. So it's about 40 amino acids that instead of like much larger molecules. Otherwise, pretty much the same, behave pretty much the same. Okay, so we've seen neurotransmitters, right? So this is what's being released at that synaptic end bulb, helping cross that synaptic cleft so that postsynaptic neuron can fire and continue that signal to wherever it's got to go, right? So we're going to talk about just a couple of neuropeptides real fast. Not a lot of them. So here's something called substance P. So basically substance P, we know that one of its jobs is to enhance the perception of pain. So if you block substance P, people report things not hurting as much or at all. Okay. Enkephalins and endorphins are basically in the same class. These things are endogenous morphine. So your body, let's see. So this term endorphin comes from endogenous morphine. It's a kind of a smash up of the words. Okay. So basically one of its main jobs is going to be to inhibit pain impulses. Okay. So when you get an endorphin rush, and you think about it, usually when you have an endorphin rush, what's going on is you're doing something that's really exciting, right? You're taking part of some sort of, I don't know, adventure or something. That is not the time that you want to stop and feel pain, right? So a classic time you'd have an endorphin rush is during your fight or flight response, right? So either you got to fight off the zombies or run away from them. That is not the time that you care that you scratched your arm. And I'm sure you guys have all had that experience. You've done something, you're out doing something, and then you kind of like next day you're like, oh man, how did I get that bruise? I don't remember hitting my arm. Because you don't, because these endorphins prevented you from paying attention to it, or caring about it, I should say. That's part of it. Right? They also will affect memory and learning. Endorphins are usually, they'll sort of give you that kind of that high, right? That it's a very kind of pleasant feeling. So obviously if something is very pleasant, you're gonna want to do it again, right? I mean, so basically it'll affect memory and learning, right? On a positive, and then also it can also get to like a negative as well, where again you hit your arm, you don't remember why or where or what happened. You're like, how did I get that bruise? So it can affect memory two ways. At the very low end, well, I should say, at some level, you remember, oh, that was a fun thing to do. I'm going to totally do that again. At the other end, you don't necessarily remember all the fine details. Okay. These do help control part of your body temperature. They're also released during sexual activity, so part of the things that makes that so enjoyable. And endorphins and enkephalins have been linked to different mental illnesses, and we're going to see this over and over again, that if you have too high a level of any of these neuropeptides or neurotransmitters or too low a level, it ends up causing different mental illnesses. And how can I put this? We'll see that different mental illnesses will have many different causes. Okay, so the end result may be the same, but you can have many different causes. This is why sometimes it's so hard to treat, because you don't really know what the underlying cause is. It could be an imbalance in this, it could be an imbalance in that, and you could be an imbalance in this, and there's really no good way to detect it. the best thing that doctors can usually do is prescribe a number of different drugs, and hopefully one of those will work. And then they can go, oh, okay, it was due to this. Another example of a neuropeptide is going to be CCK, cholecystokinin. You'll see this guy a lot when we get to 202. So it's found in the brain and in the small intestine. It's going to be your stop eating signal. It'll also regulate enzymes and contractions of the gallbladder. So that's part of the digestive function that it has. Again, you'll end up talking quite a bit about it in 202 as far as how it regulates secretions and then contractions in the bladder. So just for right now, just be aware of, what it is. All right. So neurotransmitter-wise, we have a couple of different categories. So we have amino acids, and then we have something called monoamines. So you can kind of see here just some examples of amino acids and then monoamines. So we're going to talk a little bit about each one of these and some of their functions and what diseases they've been implicated in. Again, very superficially. I guess I'll bring me back so you can kind of see me and my awesome hair. Anyway, so glutamate, one of the key things I want you to understand is glutamate is the main excitatory neurotransmitter in vertebrates. That's key. Okay, pretty much this is the gas it's the gas in the car, I guess, a gas pedal in the car. So whenever you release glutamate, it will excite the postsynaptic neuron. Okay, always. Glutamate receptors, we have 3 different classes of them. One is called NMDA, the other is AMPA, the other is kainate. When you have dysregulations in this receptor, what ends up happening is that people end up showing signs of schizophrenia. Yet not all cases of schizophrenia are due to this. However, it is one of the causes of it. Okay, so again, if this receptor is somehow malfunctioning, the brain, again, the electrical signals are off, so you get sort of schizophrenic-like responses from that. Okay. These AMPA receptors, they are going to generate very fast excitatory postsynaptic potentials, so that's kind of a key thing. So it's really important that they are going to be able to elicit that postsynaptic neuron to fire very quickly. And then when these kainate receptors are somehow faulty, a lot of times that's linked to epilepsy. You can sometimes think of epilepsy as sort of like an electrical storm in the brain. So you can imagine that if the receptor for basically the main excitatory neurotransmitter is malfunctioning, that sets off an electrical storm. Neurons are firing when they shouldn't be, and then you basically end up having like the epileptic seizures or visions or disorientation that people have. Okay. Do not fear this slide. You will not be asked this. I just wanted to show you that, so this is how complicated it actually gets. Okay, it is way complicated. There's so many other little molecular biology molecules really working together to show you what the symptoms are and like what the effects are. Even in vesicle transport, you can see there's a lot of things going on. So when we talk about very simplistic levels, this is why, because it gets crazy busy once you really delve into it. Maybe one of you guys will want to do that at some point. It does get kind of interesting, and really when we talk about a lot of the drugs, what they're trying to do is affect it at these very small molecular levels so you can have very specific effects. But that's for another day. All right. GABA, or gamma-aminobutyric acid, is going to be our main inhibitory neurotransmitter. So if glutamate was the gas, GABA is the brakes. So again, it's important regulating neuron firing. Now I know most of the time we talk about wanting things to go. We don't like things to necessarily stop. But just like in your car, you can't always just hit the gas pedal. Sometimes you have to stop, right? If the situation's warranted. And as I mentioned, like irregulations, if you're firing too much or inappropriately, you can get things like schizophrenia or epilepsy, right? So having a break is really important. The class of drugs that enhance the activity of GABA are called benzodiazepines, right? So one of the main class of drugs. So Valium, Xanax, Halcyon, these are all benzodiazepines. It's that category of drug. And one of the things that makes them very, I guess, dangerous, really, is when people are prescribed them chronically, because basically the body is very good at going, well, hey, I got plenty, because you're taking it in that pill form. So the brain stops making it itself. So basically the brain goes, why do I have brakes in the car? I don't need this. This is just dead weight. I got plenty of stopping power. It's artificial stopping power, right? So as the brain gets rid of the brakes, if you all of a sudden go cold turkey off these, what ends up happening is you end up having seizures and possibly death, actually, because you've taken away the brakes of the car, because the brain can't compensate for that quickly enough. That's why usually if someone's on some drugs, they'll usually taper them off, give you lower doses. That allows the body to kind of catch up and bring its own manufacture of that back to whatever it was at baseline levels. Hopefully that makes sense. I'm gonna hide me here. Another neurotransmitter is going to be norepinephrine. It's produced by a brain region called the locus coeruleus, also by your adrenal glands. Another name for norepinephrine is noradrenaline. But we tend to call it norepinephrine in neuroscience. The locus coeruleus in the brain has projections to many other brain regions. So what it ends up doing is that it helps increase vigilance and arousal in other brain regions. So it helps not just wake you up, but also helps you pay attention. And if levels get too high, you no longer can pay attention. Now you're kind of like, you're just not focused. You're looking at this, and all of a sudden this, and then this, and this, and this, and this, right? Which, when we think about it, is really important evolutionarily. Because think about this. At some level, you have to have, let's say you're out in real life, you're out in the middle of nowhere, in the middle of nature. And I don't know, you're going to go pick berries or flowers or something, right? So you're trying to pay attention to this specific berry or flower, whatever it is you're picking, right? So you're focused, right? Your norepinephrine levels are at nice optimal levels. You're focused, you're looking for these flowers. All of a sudden, you hear a whole bunch of twigs crunching, you hear a moan that sounds like a zombie. Well, holy cow, your norepinephrine levels are going to go through the roof, right? And what is your natural response going to be? You're going to be like, where, who, what, where, what? And you're looking all over the place. You do not care about those flowers and or berries anymore. You're going to have a very hard time even going back to that action, about being able to focus back on that task, right? Because you're so worried about what's going on over there, over there, over there. And now if you hear another sound or you think you see something, you're going to be like, what? Which is important because, if it really was a zombie attack, you want to be able to quickly respond to that, right? Unfortunately, in our everyday lives, what ends up happening with people who have anxiety and stuff like that, another kind of mood disorders, is that, zombies don't attack us really in our world. So the inability to focus on tasks at hand is now a detriment. Whereas at one point it was a benefit to our survival, in our office type workplace, it's no longer something that benefits us. Another classic neurotransmitter you guys have probably heard of is something called serotonin. Again, serotonin is implicated in a lot of different mental illnesses, some depression, anxiety, food intake, and you probably can guess this. Like, if people are depressed or anxious, that definitely affects how they eat, right? And depending on the individual, either they can't eat. I'm sure we've all been so nervous that we couldn't eat, right, at one point. Or they're depressed and they don't want to eat. So it depends. Or sometimes some people, it's the exact opposite, right? They'll want to eat even more. So it just depends on the individual. Dopamine, we'll talk a little bit more about this when we get into the brain. One of the important things is that it helps regulate motor behavior. So when you lose the neurons that make dopamine, one of the things you end up with is Parkinson's disease. Okay? And usually when people have Parkinson's disease, they'll try to give them something called L-DOPA. That is a drug. It's a precursor to dopamine. So the body can easily convert this L-DOPA into dopamine. And by that, increase again the levels of dopamine in the brain to get rid of those Parkinson-like symptoms. This works for a while, but as the disease progresses, it's not enough. And you might have to have other remedies for it. Okay. And then other dopamine pathways are thought to regulate motivated behaviors. So part of that whole depression thing, when you don't feel like doing anything anymore, like you're like, no, I don't wanna, and you're like, why don't you want to go out? You used to love picking flowers. I don't know. Now you don't want to is because sometimes dopamine is also implicated in sort of mood disorders as well. Okay. And also dopamine pathways, when these are not functioning correctly, one of the other things has been shown is that some schizophrenics actually have their dopamine pathways are malfunctioning. And again, so here's another potential cause for schizophrenia. So very different from the one we saw before. Okay. You all know what ADHD is, right? Attention deficit hyperactive disorder. Interestingly, all of the drugs for depression are somehow going to increase the norepinephrine or dopamine levels, right? Amphetamine also increases norepinephrine, dopamine, as well as serotonin levels. So what's kind of weird as far as that goes is, I had mentioned to you guys before that norepinephrine, right, if we have too much of it, you go from that nice focused picking berries, flowers thing to scanning for zombies, right, if it's too high. But interestingly, if we increase the levels even more, get past that, you go back to being focused. And in all honesty, people don't know why. They don't really understand exactly why that works. They just know it does. So you get Ritalin and stuff like that, which is, in all honesty, a lot of how some of these drugs are prescribed. They don't exactly know how it works. They just know it does work. There don't seem to be, can I put this, the side effects seem less severe than the problem itself, so they get prescribed, but they don't actually know exactly how it works. Same thing with depression. A lot of these drugs, let's go back. A lot of these drugs are going to increase serotonin and norepinephrine levels. So in some of these, what they're doing is they're inhibiting the reuptake of either serotonin and or norepinephrine. So again, if you inhibit the reuptake, the more that is left in that synaptic cleft. Okay. There are many other theories for depression, so it's not just lack of serotonin, norepinephrine levels, but these are sort of one of the most commonly, I guess, ways that people medicate or try to help depression via medications is by targeting serotonin and norepinephrine levels. Okay. I also just want to talk to you a little bit about neurogenesis. So neurogenesis is, as the name implies, like genesis is like growth, like the beginning, first growth, and then of neurons. So it's the growth of new neurons. And the idea of neurogenesis in adult humans was discovered by Liz Gould. She's a professor at Princeton University. And so basically she discovered that we do in fact have new neuron growth in at least two of our brain regions. And she discovered this somewhere in the mid-90s. So, about 20 years ago now. I guess it's 20 years ago. Yeah, 20 years ago now. And this was a very big deal because it had been stuck in the literature that once you're an adult, that's it, your brain is set and you have what neurons you have, and once they die, they're dead. But she's shown that that's not necessarily true. So basically where we see it is in the hippocampus and in the olfactory bulb. So here's the hippocampus. The hippocampus is important in learning and memory. In depression, we see that if the hippocampus is small for some reason, like it's shrunk, people tend to exhibit depression. Some people with very small hippocampuses some of them have shown symptoms of PTSD, right? So clearly proper new neuron growth is going to be really important in learning, memory, depression, PTSD. Plus, if we can figure out how new neurons grow, we can find out a way to heal damaged brain areas. Right? The olfactory bulb is going to be much more, I think, obvious to us. I mean, obviously, when you smell things, a lot of times you can imagine how you can damage that area. So you just have constant replacement of cells there, right? Just from very strong smells, I guess. And we'll talk more about, just kind of keep this individual sort of in the back of your mind. We're going to talk more about this individual named called HM in the psychology literature. He was incredibly key to our understanding of how learning and memory actually works. All right. So just very quickly, some other diseases relating back to neurons. You may have heard of multiple sclerosis. Basically what happens is that myelin sheath surrounding the neurons in the central nervous system starts to degenerate. It's an autoimmune disease, meaning that your own immune system attacks the cells producing the myelin sheath, so it attacks the oligodendrocytes. And basically it gets its name because the myelin sheath degenerates in multiple areas into plaques called scleroses. So that's how you get that. And then obviously as that myelin sheath degenerates, it'll affect proper neuron firing, which then affects function both in thought as well as muscle movements. Seizures and epilepsy, again, it's really, you can kind of think of it as an electrical storm in the brain. And again, it may or may not incorporate muscle spasms. So I think for most of us, an epileptic seizure is kind of when you see someone like really shaking and you know, kind of falling to the ground. It doesn't always mean that. We'll talk more about brain regions after this exam, and you'll find out that depending, if you have a, let me put it this way, if you have an electrical storm in the region of the brain that's responsible for you interpreting sounds, you may have auditory hallucinations. If you have an electrical storm in the part of the brain that's responsible for vision, you might either see things or you're unable to interpret that which you see. So things like that, and so it may not have anything to do with any kind of muscle spasms. Okay. And then things like rabies, right? So rabies is a virus. It's small. It can get across that blood-brain barrier. And basically what ends up happening is it causes inflammation in the brain. You have inflammation in the brain that crushes neurons. You crush neurons, you kind of, you know, you've heard of rabid animals. They don't act right. They don't walk right. They're frothing at the mouth, all sorts of other things. Because, you know, if you're crushing neurons, you can't function well. You're not thinking right. So behavior is going to be very bizarre until finally the animal just dies. All right. So hopefully that made sense to you guys. I know this is a bit of a longer lecture, and I'm sorry. I usually try to keep these under 20 minutes. This is kind of like double time. But I don't know. I think they all kind of, the topics went together, so I didn't want to separate them. Anyway, if you have any questions, please let me know. Feel free to email me as always, and I will do my best to answer your questions. All right? See you next time.